Well, thanks, Andrea, for this presentation, and thanks to Steve and Andy as well for the invitation. I'm delighted to share with you some of our recent results about how the variant Delta replicates, fuses, and interacts with the host immune system. So you all know that some SARS-CoV-2 variants emerged during the last year. The first predominant strain was the Alpha strain, which was first identified in the UK. But you are aware also that now the Delta variant is becoming predominant, and it represents, as of today, about 85% of the circulating sequence strains. This Delta variant is a fast-spreading variant. So it was first identified in India and then spread to UK, and now it represents about 99% of the cases in India, UK, and about 80% in countries like USA and France. So why is this variant spreading so efficiently? When you look at this at its sequence, it carries a number of mutations, which are depicted here, including 9 mutations in the spike. And we can classify those mutations into 3 main types. The first type are mutations which increases affinity to the receptor, ACE2, or infectivity. The Delta variant doesn't carry the N501Y mutation, which is a famous mutation present in alpha and which strongly enhances affinity to ACE2, but it carries the L452R mutation, which may enhance infectivity and also antibody escape. The second category of mutations include the P681R mutation, which is located at the S1S2 furin cleavage site, and which will enhances cleavage and fusion, and I will discuss this point in the coming slides. And finally, the third category of mutations are those which allow antibody escape, and they are present in the NTD, N-terminal domain, including the super antigenic site, as well as others like this one, the T478, in the receptor binding domain. So a few words now about fusion. You know that the fusion is very important at the early step of the viral life cycle, when incoming viral particles will fuse their membrane with the target cells. But there is another step of the viral life cycle in which fusion occurs. It's once the cells are productively infected, they may express at their surface the spike, and this spike will allow fusion with neighboring cells, and this will lead to the formation of syncytias, which are large multinucleated cells. They are easily detectable in cell culture systems, but they are also found in infected patients. I wanted to quote here 2 articles by Moro Giacca, including one in Nature, describing that in about 90% of patients who deceased with a critical disease, they were able to identify those syncytia. Their role is not fully understood. Do they allow a viral spread or viral escape from the immune system? That's precisely what we are studying in the laboratory. So now a few words about the experimental systems that we are using, and I wanted to describe the cell type, which is called U2OS, that we have been using a lot for our studies. We selected those cells because they are flat, and this allows, this facilitates imaging and automated imaging of the cells. So those cells do not express the receptor. So we stably introduce the receptor in those cells. Without the receptor, there is no, maybe I can remove this. Without the receptor, there is no fusion and no viral replication at all. But in the presence of the receptor, the cells become very sensitive to SARS-CoV-2 infection. We also introduced in those cells the GFP split system to reveal cell-cell fusion event. So this is a very simple system in which half of the cells will express part of the GFP, and the other half of the cells will express the other part of GFP. And when the cells fuse, as you can see here, the syncytia will turn will express GFP, and we can counter stain here the syncytia with different kind of dyes. So we term this system the S-fuse cells, and with this system we were able to do real-time imaging of infection and syncytia formation. So here this is a video microscopy experiment, and you can see that as soon as a few hours post-infection, the cells will fuse and generate those large syncytia, which will, after about 1 day, will end up dying, shrinking, and exploding and dying. So we use those cells for different purposes, to study syncytia formation, but also to study the neutralizing antibody response. And the system is pretty simple. We can prepare the cells, add the virus that we want to test in the presence of sera, antibodies, and drugs. And after an overnight incubation, this is a 96-well plate, and here this is one plate without any virus, and with virus you can see that the syncytia can be automatically scored, and we have an automated confocal microscope which allows to read one plate in about 5 to 7 minutes. So this is an example of the experiments we can perform, and here this is the test of 4 different sera, and we can perform serial dilutions of the sera, and you can see that, for instance, serum number 1 is barely neutralizing, whereas serum number 2 is highly active, and this is not the case here for the serum number 3. And so we can easily calculate IC50 for a large number of samples. So we were also able to isolate infectious variants, and actually we isolate the Delta variant very early in April from a traveler coming back from India, and this was a collaboration with clinicians and biologists from a hospital in Paris. So we were able to sequence the virus and to study its replication, its fusogenic activity, and its sensitivity to neutralizing antibodies in comparison to other variants that we have also isolated. So one of the first experiments and observation we made is that, and that's true also with the alpha variant, the syncytia formed upon infection, are much larger, are much larger with the alpha variant, but also with the delta variant. So it seems that those circulating and variants of concern increase their replication, or at least their ability to fuse with neighboring cells. And this was mainly due to the spike itself. Here we expressed the spike, alpha or delta spike, and we were able to quantify the syncytia, the surface of the syncytia, and it is strongly increased with both alpha and delta. And this is due to an acceleration of the fusion of the fusogenic activity of the spike. Here we can do real-time analysis of the movies that we generated. So both alpha and delta are more fusogenic than the ancestral D614G strain. We also looked at the affinity of those two strains to ACE2, so by incubating infected cells with a soluble ACE2. And there is a strong increase of affinity with the alpha strain and compared to D614G, whereas actually delta is intermediate between those two strains. So there is accelerated fusion of both alpha and delta spikes. It has also been recently shown that the delta virus is able to replicate more rapidly than the alpha virus. This is done in competition experiments in which you mix the two viral strains together, and you measure the relative replication of both strains, and there is a 4 to 7-fold increase here in Calu3 cells with the delta variant. So the delta variant seems to be more fit in a way, and it fuses, and this is in part due to the fact that it fuses more rapidly. So now, what is the sensitivity of the variant delta to antibodies? So we tested monoclonal antibodies and sera from convalescent individuals or vaccine recipients. And you all know that so far, the levels of neutralizing antibodies is a strong has a strong predictive value of immune protection from symptomatic infection. This has been shown initially in animal models, including macaques and small animals like rodents, but this is also the case in human beings. So when you have a decrease of the levels of antibodies, you will probably get more and more breakthrough, as described by Nassim in the previous talk. So we first tested the sensitivity of the different variant of concerns to monoclonal antibodies. Some of them are currently in clinical use, including the bamlanivimab. Actually, this one is no longer in use because, as you can see here, is totally inactive against Delta and as well as Beta variant, whereas the other antibodies here, those three remaining, display different kinds of sensitivity or efficacy against the Delta variant. So the variants Beta and Delta are resistant to neutralization by bamlanivimab, and both this antibody and etesevimab do not neutralize the variant Beta, by the way, as shown here. We tested a panel of 8 other monoclonal antibodies. 4 of them target the RBD, and 4 of them target the N-terminal domain, and those antibodies were generated by Hugo Mouquet at Institut Pasteur. And again, with the anti-RBD antibodies, you can see that some of them, including this one, the RBD85, is no longer active against variant Delta. And this is also the case for the 4 anti-NTD antibodies, which, by the way, are less active. Their IC50 is quite high against the different viral strains, but 3 out of 4 of them lost any activity against the Delta variant. Why do they lose any antiviral activity? It's a matter of binding affinity to the spike due to the mutation. Here, those radar plots illustrate the fact that those 12 antibodies, they all bind to cells infected with here the D614G strain, whereas with alpha, only 9 of the 12 antibodies are binding to the infected cells, and with Delta, only 7 of the 12 tested antibodies are able to bind to the infected cells. So the escape of variants to neutralization is due to a reduction or loss of binding of the antibodies. So what is the sensitivity of the variants to sera from convalescent individuals? Here, in collaboration with biologists from Strasbourg Hospital, we tested individuals who have been infected for more than 1 year. And so their anti-neutralizing activity within their sera is decreasing over time. And they lost, most of them, or a large part of them, lost any neutralizing activity against both Delta and Beta. However, when those individuals are vaccinated once with a single dose here, and this has been shown by different teams, there is a strong increase in the antibody levels, about 2 to 3 logs increase, and now all the strains are neutralized. But there is still about a fourfold decrease in the efficacy of those sera against Delta compared to alpha. And what about the vaccine recipients who have not been previously infected? So here we tested individuals receiving either Pfizer or AstraZeneca vaccines, either 1 dose here or 2 doses. After 1 dose of vaccine, the levels of antibodies were quite low and barely able to neutralize Delta. And after 2 doses, there is an increase, of course, of the neutralizing antibody levels with both vaccines. And all of the individuals were able to neutralize those variants. And again, there is a fourfold decrease in the efficacy of the neutralization with both viruses. So here, that was at an early time point, week 5 after the second dose, or week 4, about 95% of individuals are able to neutralize our neutralizers and neutralize the Delta variant. We are currently studying the evolution of the durability of the antibody response. And there is, here now we have about 6 to 7 months after the second dose, and there is a waning decrease of the antibody response against alpha, but this is even more marked with beta and delta. With beta, it's particularly marked. About 50% of the individuals are no longer able to neutralize beta, and it starts to be almost the same with delta. And this may explain why there are breakthrough infections occurring in vaccinated individuals. So what is the link with those results with the real world situation? And I wanted to quote this article that was published yesterday, well, this comment which was published a couple of days ago about the situation, for instance, in Israel, and the note is quite pessimistic, because in this country, you may know that about 80% of individuals have been vaccinated, and now they are experiencing an increase in the cases of COVID-19 infection and an increase of hospitalized individuals. And what is quoted here is that as of August 15, there were about 500 individuals who have been hospitalized with severe or critical disease, and about 60% were fully vaccinated. So the situation is quite grim, and probably a third dose will be necessary in most of the infected, in most of the vaccinated individuals. On an optimistic note, I wanted to quote also this article, which was published yesterday. This is an epidemiologic survey which is regularly performed in United Kingdom, and this was done during May to last July, and about 500 individuals were positive out of 100,000 tested swabs. And of course, you have a high vaccination rate among these individuals, but the prevalence of positivity among those unvaccinated was threefold greater than those who were vaccinated. So there is a vaccine effectiveness in the real world, and about a threefold decrease, fourfold decrease when you are vaccinated. And there is a debate about the vaccine effectiveness, and even if you get the virus after being vaccinated, it seems that the CT, so the viral loads, are reduced when you are vaccinated. Here, the CT for vaccinated participants was around 28 compared to 23 with unvaccinated individuals. That means that there is about a 2 to the 4 more virus in unvaccinated individuals. So the viral loads are about 20-fold higher if you are not vaccinated, and you will, of course, be able to transmit the virus more efficiently if you are not vaccinated. So I will stop here and conclude on what I've just shown you. So alpha and delta are more fusogenic than ancestral strains, and delta is less sensitive to sera from naturally immunized individuals than alpha. And it is now well accepted that a single dose of Pfizer or AstraZeneca does not trigger seroneutralization against delta. And the 2-dose vaccine regimen induces cross-reactivity against variants with about 4-fold lower efficacy against delta. And there is a decrease over time that we are currently studying. And the beta and delta, especially beta, escapes binding and neutralization by some monoclonal antibodies. And we are currently performing longer surveillance periods and studying also the neutralizing antibody response in individuals who received other available vaccines throughout the world. And so some of those results that I've shown you have been published in this article in Nature, and there are other articles with the same kind of results indicating the reduced sensitivity of SARS-CoV-2 variant to antibody neutralization. So I would like to stop here and to thank my collaborators. This is all my fantastic team, and I've highlighted in red the individuals who are involved in studying the fusion and replication of SARS-CoV-2 variant. and those who are being involved here on studying the neutralization of those different variants, including Timothée, Florence, Delphine, and Isabelle. And this is possible through long-lasting collaborations with clinicians and virologists from Institut Pasteur and hospitals throughout France as well. And of course, I would like to thank the patients as well for participating to our studies. And finally, I would like to thank the funding agencies. And I thank you for your attention.
Thank you, Dr. Schwartz. A very sobering presentation. We're opening up for questions. While we're waiting for people to raise their hand, a couple of questions myself. So you presented data from the AstraZeneca and Pfizer vaccine. What about single-dose vaccine like J&J?
So we are currently studying this kind of single-dose vaccines, and the levels of neutralizing antibodies. I will stop showing my screen. The levels of neutralizing antibodies are very low in our hands, in our assay, with a single dose of J&J. And meaning that my feeling is that individuals who receive only one dose won't be efficiently protected against Delta.
And the other question was that since beta seems to be even less neutralized than Delta, why aren't we seeing a significant spike in beta?
Yeah, that's a good point. It seems that there is a matter of viral fitness here. The beta strain acquired another large panel of mutations, so which enhance the antibody escape properties of the virus, but the fusogenic activity or the replication activity is reduced compared to the alpha strain, for instance. And for me, this may explain this is a kind of equilibrium or trade-off between antibody escape and viral fitness.
It's a trade-off between infectivity and antibody neutralization.
Absolutely.
Eric, you have a question?
Yes, thank you, Olivier. That's a great talk. It's kind of a follow-up on the last question, or the answer to the last question, and that is when you talk about Delta and the 9 mutations in spike with respect to the fusogenic assay, can you comment on whether other mutations in other proteins in the virus are contributing to that increased syncytia or increased transmission among human patients?
That's a very important point. We know that the increased fusogenicity that we observed was seen when we infected cells or when we only transfected the cells with the spike. So the fusogenic activity seems to be mostly due to the spike and not to any other viral proteins. But those other mutations which are present within other proteins are probably important. And that's something we would like to explore. I've seen one manuscript describing that in some accessory genes, for instance, ORF8, there are some mutations which may increase viral fitness or which may allow escape from CTLs, for instance. So it's not currently fully understood the role of the other mutations outside of the spike, but that's something important to analyze.
Thank you.
So I have since we have a little bit of time, there's another panelist that is looking for a question.
Yeah, hi, I could ask one.
If you can get a question.
Okay. Olivier, beautiful story, as you say, sobering data. So I wondered if anyone had yet information on the ability of any of the adeno vectors to be used repeatedly, and whether second, third doses of any of the adeno ones will be blunted or will be helpful.
That's a good point. What I know is, well, with AstraZeneca, you have 2 kinds of adenovirus. So you can avoid the immune response generated against the vector, but it's likely the case that you will generate anti-adeno antibodies, and probably they will be less efficient if you administrate again doses. I've not seen studies about the levels of anti-adeno antibodies after different administrations, but that's a very important point.
And I guess a related issue is whether multiple doses of even the mRNA ones will ultimately increase the issues of inflammatory responses and side effects that we live with anyway.
That's probably, yeah, that's an important point. As far as I know, and maybe Nassim would be able to comment, a third dose of RNA vaccine doesn't generate higher inflammatory or side effects than the first or second dose. So that's a good point. But regarding the other kind of vaccines, I don't know. And we also tested inactivated vaccines. We got samples from people being vaccinated with the Chinese vaccine, for instance. And in our hands, the levels of antibodies were not very high. That's all I can say so far.
Okay, great, thanks.
Question from Rodney Rothstein.
Yes, thank you for that very interesting talk. I had a kind of a two-part question, but it's all related to the same thing. Do you know anything about syncytial formation in patients who already have antibody, a good antibody response? And secondly, how about patients who have received the monoclonal treatment?
So those are good questions. So the autopsies, as far as I know, have been so far performing individuals who were infected only once. So that's not breakthrough infection. So I don't know the antibody state in these individuals. What is known is that individuals with critical disease, they mount somewhat lower antibody response. So probably, I would say that their antibody levels were not sufficient to maybe to decrease the viral spread or to decrease syncytia formation. In our hands, in cell culture systems, the antibodies are able to decrease the formation of syncytia, which is in a way normal. And this has also, well, it depends on the antibodies, but in cell culture systems, if you have antibodies which bind to infected cells, then there will be less fusion between infected and neighboring cells. So the antibodies should be active to prevent syncytia formation, but probably less active than against cell-free single round infection. I don't know if this addresses your question.
Thank you.
Olivier, I have one final question. I was a little bit confused because you showed data where the antibody neutralization was about fourfold higher in patients with the vaccine, even with the Delta, fourfold higher with respect to patients that were not vaccinated. And I was wondering how that jibes with the number that you show, where about 60% of the patients that were in the ICU in Israel were in fact vaccinated.
Yeah, that's a good point. So it is likely that the individuals in Israel who have been hospitalized have been vaccinated for a while, maybe 6 or 7 or 8 months ago. So their antibody levels are declining, and probably they reached a kind of threshold, of non-protective threshold, and which was easier to reach against Delta, because Delta requires more antibodies to be neutralized. So that's probably the durability of the vaccination, which may be an important parameter in those cases.
It would be very interesting to get data on when the vaccine was actually administered to patients that are vaccinated and hospitalized. I think that would be really.
Yeah, I've seen studies like that, and this is absolutely the case. It seems that it depends on the time of vaccination, obviously.